And it's my pleasure to introduce Deniz Saltukoglu. She works at the Global BioImaging, MBL, and she's scientific project manager. She obtained her PhD as cell biology in the University of Freiburg in Germany and continued her postdoctoral fellow in cellular immunology in the lab of Professor Michael Reff. In March 2024, she joined Global BioImaging at the EMBL in Edinburgh to lead the Imaging for All program, which provides researchers and imaging scientists from low and middle income countries with access to imaging techniques and training worldwide. Deniz today is presenting Imaging for All, supporting scientists for underserved regions to access imaging resources. Deniz, welcome.
That's fine, I guess. You can hear me? Yes. Thank you for the very kind introduction. It is my pleasure to close the Round the World session and, in fact, the whole EOE 2025 by sharing a funding initiative that follows the same global spirit. As many of you know, Imaging for All is a pilot mobility grant project that was created through a partnership between the Wellcome Trust and Global BioImaging, and that this grant's purpose is to support scientists from low-resource regions in accessing imaging technologies, expertise, and training worldwide. Officially, so this is the list of low and middle-income countries, LMICs, that is published by the OECD every year, updated. As we heard from Vivian yesterday, representing Wellcome Trust, Wellcome views bioimaging as central to discovery in health research, and accordingly, they fund progress in bioimaging, and they have an interest in understanding the barriers that affect progress in the field. So to identify these barriers, they commissioned the landscape review a few years back, I think, and this report identifies two main categories. One is infrastructural barriers, and the other is technical scientific barriers. And the latter are things like improvement in spatial resolution or spatiotemporal resolution in live cell imaging, or how to image across biological scales, or how to manage and analyze huge data sets that are being generated, and etc. However, not surprisingly, in the survey, LMIC scientists emphasized the infrastructural barriers more significantly compared to their high-income counterparts, pointing out to high instrument costs, lack of availability, therefore, of equipment, infrastructure, and expertise. They also point to inadequate technology transfer and competition for funding with high-income countries. So in order to address these very issues, Global BioImaging proposed the mobility grant with the aim of reducing barriers and democratizing access to instruments, training, and expertise. So Global BioImaging, as we all know, with its worldwide network, including both higher and lower income regions, is the perfect place to do this, giving an initiative like this the needed visibility, dissemination support, application support, so matchmaking between host and applicants, and also in the evaluation process. But in parallel, I think this initiative is also an opportunity for Global BioImaging Network to learn more about its own strengths, weaknesses, and the state of imaging worldwide. And it, of course, also helps broaden its connections as a global community. Okay, so what is this grant in a nutshell? It is it supports scientific visits of several weeks to imaging facilities or laboratories with imaging equipment, as well as shorter stays for training courses. So hosts can be located anywhere in the world, and applicants can apply with existing collaborators, or we can find them suitable hosts as long as the hosts agree to host these applicants. And the grant covers all expenses actually. And our budget in this funding period allows us to award about 120 grants from 2024 to end of 2026. So since its launch in 2024, we ran 2 open calls. We received about 200 applications from 33 different countries, a clear evidence of demand and also successful outreach. So the countries with the highest number of applications were Argentina, Brazil, and Nigeria. These 3 countries clearly stand out. And in the following cluster, we have India, Mexico, Turkey, Egypt, South Africa, Uganda, and so on. And our applicants come from 3 continents. So when we look at these numbers, we see room for growth in Asian applications. And as we heard from Marshita in this conference, I hope the initiation of IC will help increase awareness and interest in the region of Southeast Asia. And we are also very happy to think together about how we can increase I4A's outreach and uptake in Asia and anywhere else in the world. Our applicants predominantly want to visit European institutions. I guess some reason why this is so is Europe's long-standing open access infrastructure, the many training courses also offered by European institutions, and also the geographical proximity to many of our applicants. But we see many applications to European labs too, besides facilities. So especially the labs that develop specific imaging technologies. So we have here roughly a 60 to 40% split between shared facilities and individual labs. And as for intercontinental connections, exchanges, we do see regional mobility. So in the case of Latin America and Africa, that is all intercontinental connections, and we think this is a positive trend, so one that can foster sustainable collaborations. Now to the requested technologies so far. So when we look per continent, we do see some regional patterns actually. So in Latin America and to an extent in Asia, the interest is very strong for high-end fluorescence microscopy, like different super resolution techniques, light sheet microscopy, and FLIM is also high on the list there. But in African applications, the focus is more on fundamental techniques like wide field, phase contrast, confocal, so immunofluorescence, staining as sample prep. And in our second call, actually, we saw a boost in multiplexing projects coming from Africa. So I think the demand is largely shaped by the types of imaging modalities that are available locally and to some extent by what is relatively more advanced than what is so available at home. Okay, this slide shows how applicants discovered the call. We asked them, how did you hear about I4A when they applied? And the answers, I think, really highlight the strength of the network, our network. So GBI partners played a major role in spreading the word. So they informed their users, they shared it at meetings, in social media, and also in their newsletters. And also, I would like to point out to the training courses. So it seems like announcements during international training courses are particularly effective. I guess because participants are motivated to take advantage of so available opportunities. Okay, so now what we've funded so far. So among the funded activities, we already have many excellent examples. Here's a selection of some. So our courses range from fundamental microscopy to advanced topics like imaging-based spatial omics. So testimonials here. So one participant who attended a confocal course in York from Nigeria reflected, so I was so happy and fulfilled with the training I received. I thought I knew microscopy. I learned things I did not even know existed. So I think this is a really nice example of the grant's contribution to expanding knowledge and also perspective. So we have another participant going to Harvard's Micron tissue clearing workshop. So this is a grantee from Ecuador who started his lab recently there and is working on frog development. So he says, we don't have confocal microscopes and detection is complicated in large embryos, hence tissue clearing and expansion are two techniques that can help us achieve the correct resolution. So this is an example of increasing output in a creative way with the available equipment. Another one, so this testimonial comes from a participant from CNPM in Brazil. She attended EMBL's Advanced Imaging Technique course, which included a module on adaptive feedback microscopy. So the grantee found it highly relevant for the new imaging core facility being built within a high-level biocontainment environment, up to BSL-4. And the grant in this case is promoting exposure to cutting edge and relevant technology and even maybe opening avenues at institutional level. So beyond courses, I4A has supported a broad spectrum of research visits. So this is a visit from Mexico to ICFO Barcelona, where the grantee received hands-on guidance on how to build a light sheet microscope. And he will prepare a grant proposal now to construct a replica, which once operational, he says will serve the research community in Mexico and the wider region. And this was a paired application, and the second researcher was using the same microscope to image regeneration of the slug tail. And this data will contribute to a publication that is currently in preparation. This is another example of an Argentinian research scientist bringing a new sample preparation method home. So she visited the University of Göttingen to learn about how to expand her cells to study protein arrangements in single vesicles in the secretory pathway. So I find expansion microscopy particularly important as it has the potential to make super resolution accessible in low resource settings. It's an effective method at relatively low cost, and I really hope to see more projects involving expansion microscopy in the later calls. And one other project to highlight is the low-cost 3D printed microscope based on the open flexor body applied to bacterial optical nanomotion detection, which was developed at EPFL Lausanne, which reduces the testing time to about 10% of the standard method currently in use. And our grantee will use it in her antimicrobial research in Argentina, and she will promote its wider adoption in medical centers. So just a few more projects that we funded. So state-of-the-art bioimaging techniques like cryo-EM, lattice light sheet microscopy of cardiac organoids, single particle tracking, and one microscopy. And there are many others. These are just examples that I chose for this presentation. And so visits so far, so this is on the practical side, almost all visas could have been obtained. I mean, even in these difficult times, this is really good news that we can actually support visas. And the feedback of the grantees have been really positive. The application and travel process also received wonderful feedback, and this is also great for our small team consisting of me and our administrator, Susan Muchai. So she should be listening online at the moment. So as a conclusion, so I think the project has been contributing to mapping the landscape of research interest and technology needs across the region. It is telling us some things. It is telling us actually regional needs do differ within the LMIC sphere. But it is also telling us that community building efforts do have real effect, like making such a multi-actor initiative like this one possible. So the contribution of the community makes a very challenging global program look and feel very easy. And and we think the outcome can also serve as a valuable resource for focus of investment for funders. So through I4A, we hope to foster meaningful exchange between the global north and the global south, which can only be a good thing. And we've seen that technology transfer happens. So in this case, when the barriers are even slightly lowered, so that's also very nice to see. So just to finish off, a big thank you to all the parties involved in this audience, because many of you have been involved from the very beginning, whether by sharing resources, by reviewing the applications, hosting, applying, receiving the grant, or helping spread the word. And our third call will open in December. And yes, with that, I'm happy to take questions.